I'll jump over to screen sharing and we'll go ahead and get started. I do appreciate your time. I appreciate you guys being here today. I know everyone has a busy schedule, so it's definitely not easy to take the time out of your day for a webinar. And I also just wanted to be mindful. I think and recognize that we all come from different backgrounds and experiences and bring something different to the table. So if you don't mind, I thought it would be best for us to start with a broad paintbrush and narrow in onto finer details. And I'll do my best to elaborate on terminology that might be specific to the niche of genetics and/or nutrition to hopefully make sure that everyone feels like they have a well-rounded understanding of the material. So let's get started by regarding our health from a holistic perspective. Naturally, being social creatures, humans value and utilize communication. Comparably, on the cellular and genetic level, our cells communicate as well. They work together through this mechanism to articulate all of our physiological processes. This form of communication typically occurs through exchanging chemical messengers. These chemical messengers come in the form of neurotransmitters and hormones. So for example, if one cell in the body needs to deliver or have an impact on a target cell that's in another location, they can send a neurotransmitter and/or hormone through the cardiovascular system, for example, to be delivered to another cell in the skeletal system. You can compare these chemical messengers to sending a letter in the mail, with the target cell being the final destination. So our body uses these different mechanisms. I like to just remind us that even at the microscopic, the cellular, and the genetic level, what we're finding is there is a level of communication and added layers going on. Now, interestingly, nutrigenomics is the bridge. It gives us a way to connect and have a deeper understanding of how nutrition, our dietary intake, actually impacts us on the genetic level. So in order to really understand this, we need to lay down the foundations of what our genes are and what their purpose is. In general, genes are pieces of DNA. We house or protect our DNA inside of the nucleus contained within all of our cells. These segments contain critical information, and this information encodes or provides instructions on how to create particular traits, characteristics, or how to construct any of our body's thousands of different proteins. You can consider genes similar to words in a book. Different combinations of letters provide meaning to the words that we read. The entire book may hold instructions and details that can inform and disseminate information. Another analogy I like to provide is following a recipe book. Each recipe calls for particular ingredients. When combined and based upon following those instructions, they help us to create different meals. We use those recipes as our guides. Taking a look at this example on the slide, here is a gene that encodes for a particular protein that helps with, in this case, eye color, to determine eye color rather. Now we have different variants or variations of every gene, and that's one of the ways that we have the beauty of diversity among humans and across all kingdoms rather. So within our genes, we have this code, this genetic code, kind of like blueprints on how to construct a house. Well, within these blueprints, within this code system, we're able to decipher that or translate it to be able to construct any of the proteins that our body needs. And it is through this mechanism that we have this information. Now, the prior school of thought was that the genes that we're born with, that's kind of all we have. You get what you get and you don't get upset. What we've more recently started to understand is that our genes are not our destiny. While we inherit genes from our ancestors, they are not written in stone. So we're truly now starting to understand more of the interplay, and nutrigenomics is really our key to do so. So nutrigenomics is the field that bridges and connects nutrition and genetics. This discipline provides an insight for us to understand how what we eat affects our genes and how those genes then express themselves. The two are connected and thereby impact one another. Similar to our ability to turn on or off a light switch, our body has a mechanism to turn on or off particular genes. Our cells regulate this process through methylation, controlling gene expression. So this mechanism, which we call methylation, is a manner through which we can control what genes are expressed. What do I mean by gene expression? I think it warrants taking a moment to elaborate upon this. So while you have an entire set of genes, a whole genetic code housed in our DNA, protected inside of a nucleus, not all of that information is read all the time. Similarly, you might have a recipe book, but maybe you have your handful of favorite recipes that you kind of favor and select and read more often than others. Similarly, we have particular information in our body that we can utilize to construct certain proteins when they're needed. To give you an example, a typical red blood cell has an average lifespan of 125 days. After so, it's recycled and a new one is created. So in order for us to do so, our body is able to take this genetic information, decipher and or translate it, and express the appropriate proteins that are needed at the time. So you might be wondering at this point, what is the best way for us to support gene expression? Well, the answer is one that may come to mind. The best way to support this process of methylation is through nutrition, physical activity, consistent sleep, protection from environmental toxins, and stress relief. So you might be at this point wondering and starting to understand that there is an interplay between our lifestyle choices and our environmental exposure that kind of all come into play to determine the different recipes that can encode and create anything within our body. Now, our previous schools of thought focused on nature versus nurture. However, more recently, we've discovered the epigenetic influence. So there's a deeper interplay going on here. Since the human genome was mapped out about 15 years ago, researchers now have a better understanding of the roles and interplay between nutrition, genetics, and physiology. What we are discovering is that our epigenome is dynamic and it's modulated by environmental stimuli. Throughout our day-to-day basis, we're constantly exposed to different parameters in our environment. And interestingly, these have an impact upon us. Our daily choice of foods to eat contribute to our metabolic memory within our genome. Therefore, our lifestyle choices such as personalized diet, physical activity, and self-care benefit our epigenome and can decrease the risk of metabolic syndrome. Metabolic syndrome is kind of an umbrella term to encompass a group of conditions that can increase the risk of heart disease, stroke, and diabetes. Our environment may trigger or block genetic expression. So back to the light switch analogy, turn on or off particular genes, turn off ones that will keep us healthy, turn off genes that may predispose us to disease development. These influences from our environment may promote or protect us against things such as high blood pressure, elevated blood sugar, excess adipose tissue around abdominal organs, and regulate our cholesterol levels. To elaborate on the last one, cholesterol is an important molecule. It's present in every cell of the body. However, unchecked levels may build up in our arteries, causing atherosclerosis or hardening of the arterial walls, increase the risk of heart attack, stroke, and heart disease. In other words, in a nutshell, we have the potential to create health and stifle illness. And the ability to do so, or the tool to do so, comes from this understanding of nutrigenomics. How can we set ourselves up with healthy lifestyle behaviors that promote health and stifle illness? Well, when trying to understand this, we really have to take into consideration a multifaceted approach. On a daily basis, there are thousands of DNA alterations that occur in every human cell. If these DNA alterations are not efficiently repaired, our genome would rapidly be destroyed. So in other words, our diet and lifestyle are major mediating factors in this equation. Consider if you had to, by hand, copy and translate an entire medical textbook, right? Very dense information. That textbook has all of the information you need to understand something, but you are copying it by hand. Well, similarly, our DNA is copied, it's duplicated, it's translated. There are multiple processes that have to occur. And throughout this mechanism, there are alterations that happen. Well, it's not a perfect system. We have checks and balances built in, these physiological mechanisms that can correct errors. However, mistakes still happen. And so our body has the mechanism to kind of correct and notice these things. We can support this process through healthy lifestyle behaviors to help as mediating factors. For example, DNA damage is accelerated by oxidative stressors such as tobacco smoke, strenuous, so going overboard on exercise, and a diet that is high in processed foods. Conversely, diets that are high in cruciferous vegetables, things like our kales, our broccoli, our cauliflower, these cruciferous vegetables have been shown to lower the oxidative DNA damage rate in humans. Cruciferous vegetables include things that are on this slide, such as broccoli, kale, arugula, etc. The reason why these guys are so important, why I'm taking the time to point them out, is that they are typically higher in nutrients such as folate, vitamin K, and contain antioxidants. In other words, they're able to protect us in numerous manners. Antioxidants are known for their ability to slow the oxidation that damages cells. But the human body does not derive the same level of benefit from all antioxidants. So recent nutritionists have demonstrated through a study conducted by the USDA, the Agricultural Research Service, measured the plasma antioxidant capacity of study subjects following a single meal of blueberries, cherries, plums, and then juice, as well as grapes, kiwis, or strawberries. And what they found and published in the Journal of the American College of Nutrition was that blueberries, grapes, and kiwi fruit yielded the greatest increase in the plasma antioxidant capacity, while things like juices or even plums, despite their high antioxidant content, did not have the same impact upon the plasma antioxidant capacity levels. And this is probably because of other kind of competitive chemicals such as chlorogenic acid. And in other words, the antioxidants that they contain, while they are rich and plentiful, may not be as readily absorbed by humans. So something to take into consideration. We have the actual composition, the breakdown of vitamins and minerals of certain food items, but then another kind of added layer to the mystery is how well they are received by the body, by our digestive system, and if we are able to readily absorb them. So on the note of antioxidants, I included here on this slide different sources of antioxidants, and I thought it warrants kind of elaborating upon them. So when we consider antioxidants, why are they so important? They have this special binding ability in which they can attach to free radicals and render them harmless. On a day-to-day basis, our body has a plethora of different physiological processes that occur. Well, one of the byproducts that are given off, even just from typical metabolism, are these free radicals. If left unchecked, they can run and wreak havoc throughout the body. So the ability for our antioxidants to attach and bind to them is integral in helping to protect us. Keeping these in mind, consuming antioxidants on a day-to-day basis is one of the best ways that we can help to rid our body of these free radicals, which cause kind of an internal rusting in the body. Just as a car is able to rust when it's exposed to salt during the wintertime, for example, or salt from living by the ocean, our body also has this internal rusting that can occur. So our free radicals, we need to have a way to kind of keep them in check. And our antioxidants are a wonderful mechanism to be able to do so. So keeping this in mind, I always recommend going straight to the source. So actually consuming the whole food rather than a supplement when it comes to these types of items. And the reason being is because in the whole food, you have a whole encompassing benefit. So many different vitamins and minerals and fiber content that are present in the whole fruit or whole vegetable. We don't always have those same benefits when we are supplementing in this case. So when you can choose the whole food, it's typically more highly recommended, and it's also more easily absorbed by the body. We do have an absorption capacity. Now, as different researchers have been coming forth with their studies, there is some debate that remains around the relative impact of genes and the development of chronic disease. One particular study put forth by Dr. Mathers in a conference, he noted 97 genetic loci. Loci are locations, so different locations, 97 different ones that he had identified that contribute to fat accumulation. And what he found that all 97 of these different variants or variations upon the gene only explained under 3% of the body mass index variance. So his conclusion or the takeaway from these studies are that neither genes nor our diets alone entirely explain what's going on, in particular disease development. It's very complex. And I think considering that, it kind of helps to provide some insight that there is a very complex interplay in terms of genetics, an individual's environment, and everything else that comes into play, whether it be exposure to, do we have access to parks? Do we feel safe in our day-to-day basis? Do we have access to healthy food on a consistent basis? So considering this, other researchers tried to kind of take information from nutrigenomics studies and approached healthcare practitioners in Greece. They asked them, or they presented them with the opportunity to provide personalized nutrition and consultations to their patients. While 80% of the healthcare practitioners were willing to recommend a nutrigenomic approach, only 17% actually did so. And kind of taking a step back, we believe that this could be attributed to a level of comfort we haven't quite reached yet. So perhaps in the future, something that's kind of exciting is we could have more personalized nutrigenomic healthcare. So in other words, you could be able to go into a clinic and actually understand what are some of your genes that you're working with that we label as dirty genes. In other words, ones that kind of need some help and support, and what you do have working in your favor, and then give you the best approach strategy to support your own personal health. Overall, most cases of chronic diseases are not caused by mutations in one single gene, but rather through complex interactions among variants of several genes. And this layer of complexity is one of the major challenges faced in this line of research. So the next topic I'd like to cover has to do with methylation, this process that I've introduced a little bit to you. And this is the mechanism through which our body controls and regulates gene expression. 85% of methylation of this mechanism occurs in the liver. This process has to do with tacking on a methyl group, which you can see here in the image is just a carbon and 3 hydrogens attached together. But the attachment or the binding of this methyl group has the ability to turn on or off particular genes. And you might be wondering, how do you support this process of methylation? Well, the answer is one you may have predicted. It's through the right ratio of ingredients, through healthy nutrition, through adequate physical activity, sleep, protection from toxins, and stress relief. And so through our lifestyle choices, we're able to either support the process of methylation and or hinder our body's ability to do so. So through tacking on a methyl group, let's say this is a gene, for example, that encodes for the development of a particular condition or disease. If I'm providing my body the support through these healthy lifestyle choices, there is the potential for this methyl group to be tacked on and silence this unhealthy gene or this dirty gene that encodes for disease development down the line. And in this manner, this serves a protective mechanism. Now, another important character I'd like to introduce in today's story is called folate. Folate, otherwise called vitamin B9, is found in most of our leafy greens. In 1998, the Food and Drug Administration started enhancing the following foods with folic acid. Now, keep in mind, folic acid is different than folate, although similar. So the FDA started enhancing items such as bread, cereals, pasta, and rice. These were items that were very commonly consumed in the USA, and they began to enrich these items or add in folic acid. Now, the idea at the time was that the average USA consumer was deficient in this vitamin, and let's add it into these commonly consumed items such as bread, pasta, and rice in order to bring the levels back up to adequacy. Well, unfortunately, folic acid differs from folate because it blocks our body's natural folate receptors. The best way to acquire folate or vitamin B9 is through food, through actually consuming these items. And I've listed here different examples on the left-hand side, and on the right-hand side, for example, it shows you the percent of the daily value. So asparagus, for example, provides 67% of the daily value per cup that is consumed. Now, importantly, vitamin B9 works together with vitamin B12, also called methylcobalamin. Unfortunately, in the nutrition world, we not only give them a number such as B9 or B12, but we also give them a fancy name. You're welcome to use either one. They're both interchangeable. But what I want you to take away from this is that these two vitamins, vitamin B9 and vitamin B12, they work together as teammates, and they help to coordinate the methylation process. Consider these vitamins like Bonnie and Clyde. They're working hand in hand. And through doing so, they're able to coordinate the methylation cycle. Interestingly, also, we've noticed that a deficiency in vitamin B12 and or vitamin B9 may cause anemia. These two work hand in hand in the regulation of red blood cell production as well. So very important, not only in that mechanism, but also in the methylation cycle. Some other key nutrients I'll be covering include things like vitamin B12, niacin, vitamin E, retinol, and calcium. So interestingly, researchers identified these 9 key nutrients that may affect genomic integrity in various ways. So when consumed in appropriate levels, these 6 nutrients, folate, vitamin B12, niacin, vitamin E, retinol, and calcium, are associated with a reduction in DNA damage. In other words, these are helping to protect the integrity of our DNA and reduce damage that it may face or may come to on our day-to-day basis, from example, from environmental toxins. So nutrigenomic researchers contend that the real focus should be on the impact of multiple nutritional imbalances, both excess and deficiency on the genome. And so we're finding that we can study particular nutrients and be able to have a more cumbersome picture of how these either support our genetic integrity or may hinder or have an adverse effect. So what we're starting to notice is there is an intricate balance where consuming too much of something, which sometimes can happen in supplementation, they're at very high levels that the body is not prepared to handle. So we're noticing that consuming too much or too little presents adverse side effects. And it'll be interesting to see as that starts to and continues to unfold. So what does the future hold in terms of nutrigenomics? Well, what we're finding are that researchers are pushing for an international effort to study micronutrient needs based on differing genetic makeups among ancestral groups. We all come from different backgrounds, different ancestral influences, maybe perhaps have had some kind of migration or immigration throughout history. I know my family has. So all of these come into play and are factors to be taken into consideration. There is immeasurable value that lies in worldwide research from studying genomic and nutritional patterns. And as we start to understand this more, I think we can really have a deeper understanding of the true role that nutrition plays in these genomic interactions. In time, we hope to see important contributions from nutrigenomics for the prevention of many common modern maladies, including things like obesity, diabetes, cardiovascular disease, cancer, inflammatory disorders, cognitive disorders, visual functioning, and vitamin deficiency problems. Moreover, I'd like to share a quote presented by the Genome Health and Nutrigenomics Lab in Adelaide, Australia. Risks associated with the gene can be modified. depending on nutritional strategies. These complex epigenetic mechanisms are involved in aging and longevity. Nutrition has a strong impact upon epigenetic processes and therefore holds promising roles in regulation of longevity and aging. To conclude, I'll be sharing some insight from Bruce Ames, a molecular biologist at the Children's Hospital of Oakland Research Institute in California. His lab has documented a number of polymorphisms in genes that affect the binding of coenzymes, some of which are essential vitamins. With these types of evidence-based findings within the nutrigenomic framework, I believe we'll have more ammunition to convince government and public health officials to tackle the issue of vitamin deficiency around the world. The issue of vitamin deficiency is a very prevalent one that I think is not always talked about. But we hope that with this more targeted approach, we're likely to see political and economic forces fall in place to solve the problem. Although the complexities are substantial, I believe nutrigenomic approaches offer the best hope for understanding the molecular processes that maintain health and prevent disease. I'm hoping for worldwide collaboration, and through a multidisciplinary approach, we may work together to prevent disease development. I appreciate your time today. And at this point, I will stop the recording and open the floor